Hello everyone, today I will be introducing Caulerpa lentillifera using the example of the five kingdom classification in binomial nomenclature. Caulerpa lentillifera, also known as sea grapes or green caviar, is a type of green algae in the Caulerpaceae family. In 2019, Asia contributed to 97.4% of global seaweed production. This notable geographical disparity in seaweed output is mostly caused by the fact that, in contrast to other regions, seaweeds are a common component of diets in East Asia. Now, let's take a look at the five kingdom classification of Caulerpa lentillifera. Kingdom, which is Plantae; phylum, which is Chlorophyta; class, Ulvophyceae; order, Bryocides; family, Caulerpaceae; genus, Caulerpa; species, Caulerpa lentillifera. In summary, Caulerpa lentillifera plays a significant role in global seaweed production, especially in Asia, and belongs to the Plantae kingdom, highlighting its importance in both biological classification and human diet. Caulerpa lentillifera or sea grapes is actually a giant single cell with multiple nuclei, which also called as siphonous macroalgae, that has the ability to form large, differentiated thalli composed of a single giant tubular cell. We can distinguish sea grapes from other algae by acknowledging its morphological features. We can start with the appearance of color it has. They appear green due to the chlorophyll pigment in this alga. Next, morphology feature is thallus. It is highly branched. It can grow up to 10 centimeters long. Stolon in this alga help in vegetative reproduction by producing new fronds and rhizomes. The fronds arise from stolon and have a bead-like appearance due to the small spherical structure called ramuli. Ramuli are densely arranged around the fronds. The chlorophyll pigment can be found in the ramuli. Last one is the root-like structure called rhizode, which helps attachment of this alga. It is also responsible for absorbing nutrients from water and aiding the alga's growth. Caulerpa lentillifera is autotrophic, which produce their own food by autotroph. This nutritional method involves the conversion of light energy into chemical energy. Photosynthesis in this alga involves the use of chloroplasts containing chlorophyll pigments. The primary pigment, which is chlorophyll A, efficiently absorbs light in the red and blue-violet parts of the spectrum, while chlorophyll B absorbs light in the blue and red-orange parts, complementing chlorophyll A. This combination broadens the range of light absorbed, enhancing the photosynthesis process. Water and oxygen are utilized for carbon fixation during photosynthesis. Sea grapes can be found not only in the sea but also in brackish water, which has higher salinity than fresh water but lower than sea water. Brackish water areas such as estuaries and lagoons are transitional zones where fresh and sea water mix. This alga typically anchors itself in soft bottoms like sand and mud, and it also inhabits coral reefs, contributing to ecological stabilization among reef cultures. Sea grape cultivation historically began in Okinawa, Japan, and the Philippines, with the highest biomass production achieved under shade due to optimal irradiance levels. It grows best in irradiances below 100 micromole photons, an optimal temperature of 27 Celsius, and salinity of 35 PSU. Fertilizers are used to enhance nutrient uptake and biomass production. Reproduction occurs both sexually and asexually, with asexual reproduction involving fragmentation, where a piece of algae breaks off and grows into a new individual. Sexual reproduction, less common and triggered by environmental changes, involves monoequous thalli with both male and female reproductive organs. gametes are released in a mass spawning event, leading to fertilization and zygote formation, which settles and germinates into a new thallus. Mass spawning can cause significant ecological impacts such as rapid algae die-offs. Green seaweed, particularly this Caulerpa lentillifera, is one of the most abundant microalgae species and is widely enjoyed as a fresh vegetable, especially in Southeast Asia. Historically, seaweeds have been dietary staples since the 4th century in Japan and the 6th century in China, with people harvesting microalgae as early as 500 BC in China and later in Europe. Coastal dwellers favored seaweeds as a main dish or soup ingredient. However, European consumption is lower due to different dietary habits and regulation. Caulerpa lentillifera is rich in carbohydrates and dietary fibers, which are its most abundant components. Dietary fiber, a complex mix of carbohydrates and plant polymers, includes oligosaccharides and polysaccharides, along with non-carbohydrate components like polyphenols, saponins, resistant proteins, and waxes. These components can vary even within the same species. For instance, C. lentillifera has a higher carbohydrate content compared to C. racemos, ranging from 0.36% to 72.9% in its dry state. The dietary fiber content in 100 grams of dried C. lentillifera is approximately 17.5% to 36.7%, with water-soluble fiber content ranging from 2.45% to 17.21%. Interestingly, red algae tend to have a higher water-soluble fiber content at 15 to 22% in dry matter. In seaweed, soluble fiber can absorb up to 20 times its volume in water, aiding in stool bulking, shortening transit time in the colon, and potentially preventing colon cancer. In Caulerpa lentillifera, soluble polysaccharides mainly consist of glucans and sulfated polysaccharides. Insoluble dietary fibers, which are not digested in the human gastrointestinal tract, retain water in their structural matrix, increasing fecal bulk and speeding up intestinal transit. C. lentillifera's soluble dietary fiber content ranges from 15.75% to 28.98%, while its dietary fiber content is lower than other green seaweeds like Ulva reticulata. High consumption of dietary fiber, particularly fermentable fiber, is linked to increased short-chain fatty acids content in stool. Seaweed, include C. lentillifera, is a viable and sustainable protein source due to its low environmental impact and fast-growing rate. The protein content in seaweed is higher compared to common food staples like nuts, cereals, and legumes. Algae protein has the potential to supplement protein-deficient diets effectively. Among algae, red algae, which is Rhodophyta, have the highest protein levels, followed by green algae, which is Chlorophyta, and brown algae, which is Ochrophyta. In C. lentillifera, protein content ranges from 0.43% to 19.38%, varying due to geography, weather, and seasons, with higher protein levels reported in winter compared to autumn and summer. The protein quality of seaweed, including Caulerpa lentillifera, depends on the presence of quality of essential amino acids, which act as building blocks for proteins. C. lentillifera is considered to have a high quality proteins with an essential amino acid profile comparable to eggs and soy, except for tryptophan. It is rich in leucine, valine, and acetic acid, contributing to its flavor. C. lentillifera has low lipid content, ranging from 0.05% to 14% in dry weight, but is notable for its high levels of long-chain polyunsaturated fatty acids and carotenoids. It contains significantly higher levels of PUFAs compared to terrestrial vegetables, which are strong antioxidants and beneficial for preventing cardiovascular disease, osteoarthritis, and diabetes. Particularly, it is rich in linoleic acid and alpha-linoleic acid, which must be obtained through diet as they cannot be synthesized by most organisms. C. lentillifera is recognized for its potential health benefits, making it useful for medical treatments and prevention of various conditions. Caulerpa lentillifera, known for its antibacterial and antimicrobial properties, can inhibit microbial pathogens by disrupting the cell membrane, destroying enzyme systems, and altering genetic material. Its extracts have shown effectiveness against E. coli, Salmonella sp, Streptococcus sp, and Staphylococcus aureus, with minimum inhibitory concentrations ranging from 125.25 to 175.25 mg per ml. Additionally, C. lentillifera exhibits anti-cancer properties. Compounds like beta-1,3-xylene can induce cell death through chromatin condensation, PARP degradation, and caspase 3,7 activation. Research shows that ethanol hexane extract from these algae reduce cell viability and inhibit cell cycle progression in A147 glioblastoma cells, inducing apoptosis. C. lentillifera also demonstrates anticoagulant potential. Studies using rabbit plasma treated with beta-1,3-xylene showed extended activated partial thromboplastin time, indicating anticoagulant activity. Similar effects were observed with aqueous extracts, which prolong clotting time in rabbits and canines in a dose-dependent manner. Caulerpa lentillifera, also known as latok in Malaysia, is a popular seaweed in Asian countries, prized for its grape-like clusters filled with water. It is rich in vitamins, minerals, and dietary fibers, making it a sought-after sea vegetable in food and health industries. Commercial products like latok pop utilize shrink technology for extended shelf life and convenience. Sea grapes are known for reducing blood sugar levels, lowering cholesterol, promoting heart health, potentially offering anti-cancer properties, and preventing goiter. In skincare, sea grapes are used for their high mineral, vitamin, and antioxidant content, nourishing and protecting the skin from premature aging caused by free radicals. Thus, sea grape-based face masks provide beneficial effects for users. Sea grapes, also known for their high water content, effectively hydrate the skin and contain amino acids and essential fatty acids that maintain the skin barrier and moisturize the skin. Overall, sea grapes offer numerous benefits to humans due to their rich mineral, vitamin, and antioxidant properties, making them highly popular in consumption. Commercialized products derived from sea grapes, such as latok, cater to various consumer demands for their health and skin care benefits.